The discovery of the structure of DNA was a monumental moment in science. The entire field of genetics owes their gratitude to the work Watson, Crick, Wilkins, and Franklin did to uncover the true form of DNA. It started when a Czech monk named Gregor Mendel observed the inheritance of different traits of pea plants through breeding, showing through his research that traits were passed along in packages called genes. Through his work, he established the rule of independent assortment and the law of segregation, generating a general idea of how genetics worked at the time. 2 years later, Friedrich Miescher, a young Swiss physician, was the first to isolate a strange new substance called the nuclein from the nuclei of cells. Today it's referred to as the nucleic acid that makes up DNA and RNA, but nobody had yet known of the relationship between nucleic acids and genes at the time. After Miescher's discovery, scientists Phoebus Levene and Erwin Chargaff strengthened his discoveries with their own ideas. Chargaff discovered that nucleotides bonded with their corresponding pairs, adenine with thymine and cytosines with guanine. This is classified by their hydrogen bonds, whether the nucleotides are pyrimidines or purines. Chargaff's rules later proved invaluable while trying to come up with DNA structure. Two groups of scientists were the leaders of DNA research in the early 1950s. James Watson and Francis Crick were both scientists searching for the truth at Cambridge University. They used ball and stick models to try to figure out the structure, trying to find out all of its possible configurations. But even with Chargaff's rules, they were stuck, and all of their efforts seemed futile. Meanwhile, Rosalind Franklin and Maurice Wilkins were researching DNA at King's College in London. They used X-ray diffraction to uncover DNA's hidden secrets. The X-rays showed a shadowy picture of DNA structure from how the rays bounced off of the DNA. Franklin was a shy and private scientist who suffered from the patronizing attitudes that were delivered upon female scientists in the time. Despite her secretiveness, her partner Wilkins showed off their findings to Watson in January 1953. See, Watson and Crick were desperate for help, and after the countless attempts at trying to recreate DNA structure with models, they couldn't figure out how it was done. The data from the machine revealed DNA structure as a double helix with its black axis. With Franklin's data and Chargaff's rules, they finally had what they needed to crack the mystery behind DNA structure. Watson reportedly said, "The instant I saw the picture, my mouth fell open and my pulse began to race." Watson and Crick became the first to propose that the DNA molecule was made up of two ladders of nucleotides paired up and intertwining to form a double helix. The structure was officially announced in the April 1953 issue of Nature and explained how the DNA molecule could replicate itself with such great accuracy. For their work, Crick and Wilkins and Watson received the Nobel Prize in 1962. Because Franklin died from the radiation of the machine she worked with, she unfortunately could not receive the Nobel Prize because it is not awarded posthumously, meaning she cannot receive it since she is dead.
Created using Powtoon.